This is Microbiology Lecture 8. This is going to be on biotechnology. This chapter corresponds to the Bauman Chapter 8 and the Tortora Chapter 9 in the text. So biotechnology is just the use of microbes to make practical products. This can be good or this can be used for bad things. When we talk about recombinant DNA technology, it's sometimes abbreviated as rDNA. This is the intentional modification of the organisms by both natural and artificial means. It's sometimes just referred to as genetic engineering. Here we're using artificial selection to select for desirable traits for our own purposes. A clone is just a group of genetically identical individuals. There's three main goals of genetic manipulation. One would be to eliminate undesired phenotypic traits. Another would be to combine beneficial traits of two or more organisms. And the third would be to create organisms that synthesize products humans need. So this third one we actually do quite a bit already. When we look at the tools that we use for the recombinant DNA technology, mutagens are important. So while mutagens may be something that you try and avoid for cancer risk or to damage your own DNA, they do serve a purpose in the lab. These are used to induce changes in the genome, and then you can select for cells that are going to have the characteristic you consider beneficial. With site-directed mutagenesis, it's more targeted. You're looking to make specific changes in a gene. Reverse transcriptase is an important enzyme. We got this from the HIV virus. It creates a flow of genetic information in the opposite direction of the conventional transcriptase. So what this will allow you to do is use RNA and make a DNA from it. We call that your cDNA or complementary DNA, where you use that RNA template to make the cDNA. It's easier to isolate the desired genes on cDNA rather than an mRNA because you can have more copies of it. In eukaryotes, the mRNA has introns that are going to be removed, so it can be inserted into the prokaryotes to make eukaryotic proteins like insulin. And the way we create this is using the reverse transcriptase. So it's a way of getting the genes already cleaned up, letting the eukaryotic organism do the work of cleaning up the genes and just taking that final product that we have at the end. Synthetic nucleic acids are a useful tool as well. We can produce DNA or RNA in a cell-free solution. The difficulty with these is knowing what the sequence is of the gene. So determining what amino acids are in the gene and then the sequence of it is the tricky part. It's not necessarily actually making it that is tricky. So some uses for this, we use it to find the genetic code of organisms to create genes for specific proteins. We use it to synthesize RNA and DNA probes that we will use to locate specific sequences, synthesizing antisense nucleic acid molecules. This, we already know these can bind and interfere with genes in the mRNA molecules to act in a regulatory function, and then to synthesize PCR primers. Probes are a really big part of DNA technology. With a probe, what you have is a nucleic acid that has a specific sequence, and it's labeled so you can detect their location. These are short segments of single-stranded DNA, and what they can do is they can bind to the complementary DNA. So if you're looking to see do you have DNA from a particular organism, you can use a probe for that organism, and when you put it in the sample, if the probe sticks to that matching complementary DNA, you know you have what you're looking for. And that's going to be the big principle behind a lot of the tools we do with genetics. Restriction enzymes, these are used to cut DNA molecules. They can be at multiple locations, and they're going to cut where there is specific and palindromic nucleotide sequences. One way of cutting is to have blunt ends. Here, the restriction enzyme is going to cut both strands of the DNA in the same place. With blunt ends, it's not going to be overly picky about what it will attach to. If you make a cut with sticky ends, your restriction enzyme is going to make a staggered cut. These are not directly opposite to each other, so this can be used to join two different DNAs cut by the same restriction enzyme because each will make a complementary piece. Vectors are going to be nucleic acids such as viruses, transposons, and plasmids. These are used to deliver a gene into a cell. The goal is to insert a useful gene into the cell, and a vector is just a tool to get that in there. Some of the useful properties of vectors: we like them to be small enough that you can manipulate them in the lab. They need to survive inside the cell. They need to have a recognizable genetic marker so we know when we've actually successfully put the vector in. You can ensure genetic expression by providing the required elements like promoters, and then they should be capable of self-replication inside the cell. Shuttle vectors are capable of surviving in several different species. This way we can move a cloned section of DNA sequence among different organisms. When you're using vectors, a restriction enzyme is going to be used to cut the DNA and the vector into fragments that are going to have sticky ends. By using the same restriction enzyme, their sticky ends should be complementary to each other. The enzyme ligase will anneal the fragments into a recombinant plasmid. The plasmid is then inserted into a bacterial cell, and the bacteria is grown on a media that will select for the cell that has the plasmid. Gene libraries are collections of bacterial or phage clones. These will contain a portion of the genetic material of interest. A lot of these are actually commercially available now. So PCR, or the polymerase chain reaction, is a procedure that's been around for a while now. It's used to amplify DNA in vitro. It's a repetitive process that's going to separate and replicate the strands. So during the denaturation process, you're going to heat the sample up to 94 degrees Celsius. This will separate the two strands of the target DNA. During the priming process, you add a mix containing excess DNA primers that are complementary to sequences near the end of the DNA. DNA polymerase and an abundance of the 4 bases are added, and then it's cooled to 65 degrees Celsius, allowing the double-stranded DNA to reform. This will provide a 3' hydroxyl group that's required for synthesis. During the extension phase, you raise the temperature up to 72 degrees Celsius and increase the rate that the DNA polymerase replicates each strand. So this cycle can be repeated over and over and over. After 30 cycles, you're able to produce over 1 billion identical copies. This can give you enough genetic material to work with to do other tests. So back in the 90s, this was actually done by hand, where you would take and move the sample into different temperature water baths, and it was actually a pretty lengthy process to have to do that. Now we have thermocyclers, which are machines that are going to automatically perform PCR, and you can get over 1 billion copies within just a few hours. So here's an animation of the process. Again, I find these animations really helpful, so I recommend watching them. In real-time PCR, it's a variation of PCR. Here you can actually measure the number of DNA sequences in a sample. This can be used to monitor the progression and effectiveness of treatment. You can add a dye that fluoresces when it's bound to double-strand DNA and use a photometer to measure the amount of fluorescence. So it provides us a way of measuring or quantifying the amount of DNA. So when you're trying to do clone selection of recombinant cells, you can use the probes that are going to bind specifically and exclusively to the complementary nucleotide sequences with a radioactive or fluorescent label so that you can identify where they are. Separating DNA has been done using gel electrophoresis. This separates the molecules based on an electrical charge, size, and shape. DNA has a negative charge, so it will move towards the positive electrode through an agarose gel. DNA is digested with restriction enzymes, and that's going to make thousands of little fragments of various sizes. These are called the restriction fragment length polymorphisms, or RIFLEPs for short, and they will separate on a gel electrophoresis. In the southern blot, this will begin with gel electrophoresis. Then you will transfer the DNA to a less delicate nitrocellulose membrane. and fluorescent probes are used to localize the DNA sequence of interest. This will create a fingerprint. So this was developed by Ed Southern in 1975. The northern blot is a similar technique that's used to detect specific RNA sequences. So DNA microarrays, these are going to be made up with either single-strand DNA, either genomic or cDNA that's been immobilized on glass slides, silicon chips, or nylon membranes. These are made with robots. They deposit the PCR-derived copies of hundreds or thousands of DNA sequences in very precise locations. So single-strand fluorescently labeled DNA samples are then washed over, and those samples are only going to adhere to the complementary DNA sequences. We can use this to monitor gene expression, diagnose infections, or identify organisms in an environmental sample. So the microarrays are also commercially available. When we want to insert DNA into the cell, we have to have a way of getting it in there. There are a few ways that we can use other than using the vectors. One is electroporation. Here an electrical current is put to the used and puts a microscopic hole in the cell membrane. You can do protoplast fusion. So remember, the protoplasts are cells with the walls removed. When the protoplast encounters another protoplast, the cytoplasms fuse to form a single cell that's going to have genomes of both cells. Another way is injection using a gene gun. It's powered by compressed gas or a blank .22 caliber cartridge, or it is sometimes done using microinjection using a micropipette. So there are many applications of recombinant DNA technology. We're just going to be looking at a few of them in here. So genome mapping is a way of locating the genes on the nucleic acids. And one way of doing this is using restriction mapping. This maps the relative location of genes. You compare the DNA fragments that result from doing the cleavages by several different restriction enzymes. So this will determine each fragment's relative location to the other fragments that are cleaved. And then their sizes are determined by gel electrophoresis. So this isn't actually going to sequence it, it's just going to give you relative locations of genes, which can suit a different purpose. This is an animation here showing restriction enzyme mapping. FISH is the fluorescent in situ hybridization. This uses the fluorescent probes to hybridize with complementary targets in an environment. These can be used in clinical samples or in biofilms. DNA sequencing has changed over the years. It began when Frederick Sanger used this modified version of 4 different DNA nucleotides that would terminate DNA replication. And then he examined the numerous fragments to determine the sequence. This was called the Sanger method. Before long, this was improved. And what they did was they started incorporating 4 different fluorescent dyes, a color for each nucleotide base, and then automated sequencing that would determine the sequence of the codons. With next generation sequencing, the DNA synthesis is temporarily stopped after adding a single modified nucleotide, and each of the 4 kinds of nucleotides, the A, T, G, and C, will fluoresce a different color specific to that base. This allows us, at this point, to sequence about 4 billion nucleotides bases per day. Genomics is the study and analysis of the nucleotide bases of genomes, and it's used to cleave and map them on a gel. We now use cDNA, which is going to be faster. With functional genomics, we try and study what the gene's products do. This is done using a gene knockout, where this one single gene is removed and the rest of the genome is intact. We then will examine the phenotypic analysis, and that will reveal the function of that gene. So some of the pharmaceutical, therapeutic, and medical applications of DNA technology. One thing is protein synthesis. We can insert synthetic genes for things like insulin and interferon. This is a good way to have bacteria make these substances for us. It's safer because you don't have any fear from getting infection or contamination from the donor. It's also less expensive than the natural occurring counterparts. So vaccines, we can make subunit vaccines. This will allow you to insert antigenic polypeptides to make the proteins. As long as you have the antigen, it can stimulate immunity without actually injecting the organism. Subunit vaccines have already been used for hepatitis B. They're researching introducing antigenic proteins into common fruits and vegetables as a means to vaccinate people, or using recombinant plasmids. DNA vaccines, these are going to usually use circular plasmids with a gene encoding a viral protein. Genetic screenings, so these will use microarrays to screen for the presence of a gene causing disease. With DNA fingerprinting, here you identify individuals or organisms by their unique DNA sequences. This plays a role in paternity testing and forensics. With gene therapy, this is still in research, but the goal is that you would have missing or defective genes replaced with normal copies. Gene silencing, this is a natural process in eukaryotes. It serves as a defense against viruses and transposons. You can have your small interfering RNAs and mRNAs, which are destroyed by your RNA-induced silencing complex, or using RNA interference. So it's a way of regulating genes. There's also a role for medical diagnosis. You can use PCR and microarrays with genetic probes. These are being used more and more. They're rapid and they're highly effective. This is actually used for almost all viruses and many bacteria for detection now. With the Human Microbiome Project, the goal is to identify and categorize all the different species that live in and on the human body. So in the last few years, we've come to the realization that these microbes actually play a much bigger role in our health and our body function than we've given them credit for. Using shotgun sequencing, small pieces of a gene of a free living cell are sequenced, and then the sequences are assembled with a computer. The gaps need to be found and sequenced, but this does allow us to study genomes of microbes that have not yet been cultured. With metagenomics, this is the study of genetic material taken directly from environmental samples. What has come about as a result of this is bioinformatics. It's the science of understanding the function of genes through computer-assisted analysis. We've had to go to computer-assisted analysis because of the very, very vast amount of information. With proteomics, this is the science of determining all the proteins expressed in a cell. With reverse genetics, this is an approach to discover the gene function from a genetic sequence. This is attempting to connect the given genetic sequences with specific effects on an organism. So this is trying to be able to see what the genes do by looking at what certain sequences do, that maybe we can extrapolate that information. With xenotransplants, you're going to transfer animal cells, tissues, or organs into human tissue. There is the potential to be able to inject a gene into an animal and have them grow human tissues or organs. The benefit to this process would be that they would not be rejected. So this is still not really done. It's still something being researched. The biomedical animal models, these use recombinant DNA to make mouse tissue more human-like. It makes them more useful models because humans are not necessarily good test subjects. If we can make mouse tissue more similar to humans, we can get more accurate testing. So there's a lot of agricultural applications of recombinant DNA technology. It's been used to produce transgenic organisms or recombinant plants and animals that have been altered for a specific purpose. They're also called GMOs, or genetically modified organisms. So these are actually used widely throughout society. You actually have to make a pretty serious effort to avoid these things. So one example is herbicide resistance. Here you insert a gene from Salmonella that conveys resistance to glyphosate or Roundup. We've made them have salt tolerance. Excessive salt in the soil will decrease the amount of farmland. By inserting a gene for salt tolerance, you can use the salty oil, and the organisms can actually remove the salt to restore the soil. Giving them freeze resistance allows us to have them grow a greater portion of the year. There's a strain of Pseudomonas that has a polypeptide that prevents ice crystal formation. This has been sprayed on the crops. Pest resistance, Bt toxin from Bacillus thuringiensis. This kills insects. This has been inserted in a variety of crops. We've also modified them to change the nutritional value and yield. Bovine growth hormone given to cows is grown in bacteria. We've been able to increase the shelf life. There's been a gene to add beta-carotene to rice. With forensic microbiology, this can use genomics to trace disease outbreaks, can be used to identify DNA fingerprints or RIFLIPs in court. It can trace things back to the source. With nanotechnology, this would involve using molecule-sized robots or computers to detect contamination in food, disease in plants, or bioweapons. So anything with this technology does tend to bring about some ethical concerns and safety with the recombinant DNA technology. Just some of the questions are, is it ethical to raise animals solely to grow human tissue? One of the concerns is we don't really know the long-term effects. The gene transfer that we do may end up in organisms that we don't necessarily want it to grow in. There is the potential for creating new pathogens. This technology can be used for bioweapons. We may create new proteins that we don't really know how to deal with. With genetic screening, should it be routine or not? Who pays? Who knows the results? So at the time that I'm recording this lecture, the FDA had actually just the other day approved the first direct-to-consumer genetic screening. Should we be able to choose the sex of a child? So we know that that does have some potential problems where there was selection for sex in certain countries. They would end up, they didn't do genetic testing for it, but they would just basically get rid of their female children. But there were villages that have now got large numbers of adult males and they've got no females. So then when it comes to patents, who can make the patent? What can they patent? All of those things are still things we need to think about and determine what's going to work for us as a society.